Our next presentation is a virtual presentation by Dr. Shalena Naidoo, and this is a longitudinal assessment of the impact of antiretroviral therapy and HIV-1 associated inflammation on neurocognitive outcomes in perinatally infected children in South Africa.
Good afternoon, and thank you for the opportunity. My name is Shalena Naidoo. I'm from Stellenbosch University in Cape Town, South Africa. I'll be talking about a longitudinal assessment evaluating the impact of antiretroviral therapy and HIV-associated inflammation on neurocognitive outcomes in perinatally infected children. To provide some context to the interplay between HIV, the central nervous system, and the immune system, we know that within days of primary HIV infection, the virus can enter the central nervous system and result in HIV-related neurological manifestation. HIV does not infect neurons of the central nervous system, but it infects glial cells that support and protect these neurons. In addition to this, the HIV results in inflammation that leads to further damaging of the brain and the spinal cord. Immune surveillance and maintenance of homeostatic conditions of the central nervous system is habituated by both innate immune cells and specific subsets of T cells. HIV can therefore deflect the central nervous system barriers through either transcellular, paracellular mechanisms or by the Trojan horse effect, where infection of the central nervous system results in the infiltration of leukocytes and leads to HIV-infected monocytes and T cells that are able to cross the blood-brain barrier. So in the context of viral persistence, there is a drive towards activation of microglia and astrocytes, therefore inducing pro-inflammatory cytokines, and further drives neuronal injury, blood-brain barrier damage, brain atrophy, and ultimately leads to HIV-associated neurocognitive disease, or HAND. Now further to this, macrophages may harbor latent HIV reservoirs, HIV-1 viral proteins may activate nearby macrophages, and this leads to a whole host of cytokines and chemokines that are released and drive the process of localized neurological. Now, children are 3 times more prevalent to developing neurological disease compared to adults, with 75% of cases occurring within the first 2 years of life. The vulnerability of the developing brain, specifically in perinatally HIV infection, presents more frequent central nervous system opportunistic infection as a result of HIV immunosuppression compared to adults. Now, in the absence of combination antiretroviral therapy, between 20 and 50% of children with perinatal HIV infection will develop HIV-associated encephalopathy. This is due to complications associated with uncontrolled HIV infection. Now, PHIV is strongly associated with reduced brain volume and chronic immune activation and inflammation. In a South African study, cortical and subcortical structural impairments in children with perinatal HIV infection was documented to be the major neurological manifestation that is associated with long-term neurological impairments. However, even in the setting of early initiated combination antiretroviral therapy, sustained biological control, and good clinical follow-up, children with perinatally HIV infection still show compromised and worsened neurodevelopmental outcomes when compared to their uninfected counterparts. There are 3 categories of HAND that has been defined within the general HIV population in order of increasing cognitive and functional severity. These include ANI, asymptomatic neurocognitive impairment, MND, mild neurocognitive disorders, and HAD, HIV-associated dementia. There are many risk factors associated with HAND in people living with HIV, and these include CD4 T cell counts, the timing of diagnosis, native T cell counts, duration of HIV infectivity, age, and co-infections. Now, the incidence of ANI and MND remain relatively high in biologically suppressed individuals, and this leads to deficiencies in compartments of the memory, learning, and attention functions. Now, a number of driving factors exist for HAND during biological suppression. These include persistent inflammation, the low-level viremia viral replication that occurs, gut damage or leaky gut syndrome, possible epigenetic factors and co-infections. These may include CMV or hepatitis C, which are common within people living with HIV. The identification of biomarkers that may predict and provide further insight into neurocognitive impairment in vulnerable populations is considered a key research focus. There is a need for methods to be developed that reliably predict hand before onset and accurately diagnose cognitive impairment, and also screen for changes in cognitive performance. Clinical and immunological biomarker associations with neurological outcomes in children with perinatal HIV infection are limited. Not only do current studies not examine a wide range of clinical and immunological biomarkers and their resultant relationship to neurodevelopment, but also do not evaluate the change in these biomarker levels that occur longitudinally in relation to disease progression, different therapy regimens, and long-term biologic. So this research study gathers the findings of two key sub-studies, immunological and neurodevelopmental, of the larger cohort study. Participants originated from the well-known and well-characterized SHARE randomized controlled trial, where study participants were randomized into 3 study arms, including ART deferred or delayed, ART initiated early and then interrupted at 40 weeks, and ART initiated early and interrupted at 90 weeks. The study design adopted focused on both longitudinal and cross-sectional. Neurodevelopmental assessments were done by Professor Barbara Lawton from Stellenbosch University, where the Griffiths Mental Developmental Scales administered at 11, 18, 30, 42, and 60 months of age, and it included assessments of locomotor, personal social, hearing, language, eye-hand coordination. The Berry-Buttanica developmental test for visual motor integration was implemented at 5, 7, and 9 years of age. We evaluated 40 immunological plasma biomarkers by Luminex multiplex assays, or ELISA. This biomarker panel spanned through chronic inflammation, monocyte macrophage activation. It evaluated cytokine and chemokines related to T-cell regulation, as well as gut-related inflammation and overall chronic inflammation. We evaluated total HIV-1 DNA by using a sensitive qPCR adapted for subtype C and targeted the integrase gene. The figure on the right represents the time points measured for the neurocognitive assessments versus the immunological assessments. We see that there are tests 1 to 8, and these are specific for the neurocognitive assessments, and then immune biomarker measurements were done before therapy initiation within the first 0 to 12 weeks, and then biomarker analysis at 12 to 24 months, and then at 4 to 5 years, and then at 8. So a brief overview of the sampling outline, which spanned between longitudinal and cross-sectional study designs. Neurodevelopmental outcomes were measured in 139 participants between HIV-infected, HIV-exposed uninfected, and HIV-unexposed uninfected children. For correlation analysis between baseline biomarkers, clinical, biological, and other immunological biomarker parameters, these were measured from 11 months to 9 years of age in 45 HIV-infected children only. Another longitudinal assessment was initiated to evaluate a correlation between clinical, biological, and immunological parameters with neurodevelopmental outcomes. This was done in 69 participants of only HIV-infected. A cross-sectional analysis was done to evaluate the relationship between clinical, biological, and immunological HIV-infected, HIV-exposed uninfected, and HIV-unexposed uninfected children at 8 years of age with neurodevelopmental outcomes at 5, 7, and 9 years of age. In this study, we stratified our study participants into 4 key groups. Group 1 depicted delayed therapy, Group 2, early continuous therapy, Group 3, early therapy interrupted at 40 weeks, and Group 4, early therapy interrupted at 90 weeks. The groups were relatively similar, except we could point out that for Group 4, we only had 3 participants. Here we present the baseline HIV and immunological parameters for the HIV-infected group. Here we show that the time to therapy initiation was 9 weeks within the delayed arm and approximately 2 weeks within the rest of the study groups. Time to viral suppression was significantly higher within group 3, which was the early therapy initiation and interrupted at 40 weeks. And then looking at a CD4 percentage at birth, we see that group 2, the continuous therapy group, had a CD4 percentage of 28.5%, which was relatively low compared to the other groups. In terms of the 8-year follow-up parameters, the groups remained relatively the same when evaluating parameters of CD4 cell counts, percentages, and CD8, as well as CD4-CD8 ratios. Then just to provide a snapshot of all our HIV-infected children groups at 8 years of age compared to HEUs and HUUs, we see that the CD4 count and percentages were relatively the same across all 3 groups. However, differences did exist between CD8 cell counts and CD8 percentages, as well as CD4 and CD8 ratios. On the right, we show CD4, the longitudinal snapshot of longitudinal percentage CD4s, where we can see that all our groups across each therapy arm in the HIV-infected children group showed relatively similar patterns. Similarly, with cell-associated DNA, we see a progressive decline in CAD across all the study groups evaluated within the HIV-infected group. In terms of neurodevelopmental outcome measures for this particular study, we focused on locomotor, the BMI raw scores, and then the general Griffith scores. We noticed that the delayed therapy arm showed lower neurodevelopmental scores from 11 to 42 months, and following this initial developmental delay, there was a notable recovery by 60 months of age. We also noted that therapy interruption had minimal impact on the overall neurodevelopmental scores. Now, evaluating the correlation between immune biomarkers at baseline, that is before therapy was initiated, and the neurocognitive scores from 11 months to 9 years of age, we found based on our heat map analysis that there were significant positive associations between pro-inflammatory biomarkers related to IL-1 beta, soluble CD14, soluble CD163, IL-18, LBP, and the early locomotor and general Griffith scores. Now, these biomarkers indicate an innate immune activity of monocyte and macrophage activation and PAMP stimulation, pathogen-associated molecular patterns, and this may seemingly depict a neurological protective response that is happening. In contrast, we found negative associations between IL-1RA, IL-6, MCP1, MIP1 alpha, interferon alpha, IL-17F, IL-12, MIP1 alpha, as well as TNF beta. Now, these biomarkers may serve as early predictors of late neurodevelopmental outcomes, whereas IL-13, interferon alpha, IL-6 can predict both early and late neurodevelopmental parameters. Here we show associations between baseline clinical parameters that can predict neurocognitive outcomes between test 1 to 8. We find that early measures of HIV CAD were significantly associated with locomotor and general Griffith scores. In addition to this, we found both positive and negative correlations between clinical parameters and early and late neurodevelopmental outcomes. These clinical parameters included time to viral suppression, gestation, birth CD8 percentage, CD8 count, time to ART initiation, as well as baseline CD4. On this particular slide, I would like to highlight the analysis achieved following association studies where we measured the change in biomarker levels with change in neurodevelopmental outcomes. Here we saw significant positive associations between pro-inflammatory cytokine IL-18 from baseline to 8 years and the association with BMI scores from 5 to 9 years of age. In contrast, we found significant negative associations between MCP1, TNF-alpha, IL-18, and LBP. At pre-adolescent age of 8 years, biomarker associations with neurodevelopmental outcomes showed significant positive associations with intestinal fatty acid binding protein at 8 years and BMI standard draw scores. In contrast, we found significant negative associations between IL-17F, IL-12, IL-13, interferon gamma, MIP1 alpha, TNF-beta, and interferon alpha. These observations show an interplay between inflammation and protecting the blood-brain barrier, thereby contributing to preserving some mechanisms of brain function within perinatally HIV-infected children. Our study was able to delineate the mammological predictors of neurodevelopmental outcomes. As early predictors, we found that soluble CD14, soluble CD163, LBP, IL-18, IL-13, interferon alpha served as good predictors for neurodevelopmental outcomes, whereas interferon alpha, IL-17F, interferon gamma served as good late predictors of neurodevelopmental outcomes. Spanning these all together, we found that a host of immune biomarkers, including LBP, IL-18, TNF-alpha, and MCP1 served as longitudinal predictors of neurodevelopmental outcomes, as well as evaluating the change in biomarker versus neurodevelopmental outcomes in perinatally HIV-infected children. Further to our analysis, we decided to evaluate perinatal pro-inflammatory biomarkers as predictors for basal ganglia or volumetric disturbance at 5 years of age within HIV-infected children. We measured the impact of early inflammation on subcortical structures in the CNS. So at 5 years of age, the Sher children underwent brain imaging scans, and we selected for structures within the basal ganglia. These included the caudate head, the nucleus accumbens, the putamen, the globulus pallidus, the corpus callosum. As we see on the right-hand side with the clustering group figure, we found that lower subcortical gray matter values have higher immune biomarker values associations. There were 3 unique subject grouping identified in the analysis, which is represented by the dashed line in the figure on the right. The corpus callosum values appears to group with higher concentrations of the blood biomarkers. We found that IL-10 was negatively associated with the bilateral putamen and globulus pallidus. There were 7 pro-inflammatory markers that positively associated with larger gray matter structures. We also found that higher LBP and MCP associated with decreased corpus callosum size, which was the opposite of the finding of the gray matter response to pro-inflammatory biomarkers. The degree of initial immunological response in early perinatal infection has significant long-term impacts on subcortical central nervous system structures. And so this impact of the severity of initial HIV infection has persisted on ART. In this slide, we show a summary of our findings, which is a separation between the gray and the white matter, as well as a listing of the immunological biomarkers, which were either pro or anti-inflammatory. The upward arrows indicate a positive association, whereas the downward arrows show a negative association. So within the gray matter, we found positive associations between RANTES, GM-CSF, eotaxin, and certain structures of the gray matter. These were mainly pro-inflammatory of nature. We identified 8 immunological predictors of subcortical volume. These included IL-10, TNF-alpha, RANTES, LBP, and MCP1. With regards to the white matter, we find negative associations between LBP, MCP1, and the corpus callosum. In conclusion, we found that neurocognitive outcomes can be predicted by early immunological, biological, as well as clinical parameters. We concur that early initiation of ART is significantly important for mitigating immune activation and inflammatory response, which then impacts the neuroimmune relationship and brain development. The degree of initial immunological response and early perinatal infection has significant long-term impact on the central nervous system and subcortical structures. Pro-inflammatory markers associated with larger gray matter structures and anti-inflammatory markers are associated with decreased structure size. Our data also highlights the important role of early immune response to HIV in early and late neurodevelopment and provides a foundation for future research. There's also a role to further explore the gut-brain axis relationship within this population. I would like to acknowledge the contributors to this presentation and research, as well as the funding bodies. Thank you for your time, and I'm willing to take any questions.
Thank you very much.